Hello and welcome back. This lecture we're going to talk about a very important chemical pathway in biology that is used to create energy, cellular respiration. One thing I like to note about this particular chapter, this material is hard. In fact, when I was your age, I thought this was the hardest chapter in Bio 111. With that said, I will do my best to break the material down so that you understand it. I do highly recommend that you read the chapter. If there was ever a chapter to read, this is it. Also, I'll be posting videos of some of the chemical reactions as well. It would be in your best interest to watch those and to take your time with this material. Do not rush through it. With that said, do not be intimidated by the information. I know you can do it. With that out of the way, let's dive in, shall we? This lecture will focus on cellular respiration. Before we get to that, we need to learn about ATP, oxidation reduction, and then finally, 4 steps of cellular respiration. It's a lot to go over, so again, please take your time with the material. Energy, as we learned in Chapter 2, is stored in chemical bonds. Energy is released and transformed by metabolic pathways. Chemical energy that is available to do work is called free energy. There are 5 principles that govern metabolic pathways. As we will see, cellular respiration is no exception. First, chemical transformations occur in a series of intermediate reactions that form a metabolic pathway. Second, each reaction is catalyzed by a specific enzyme. Third, metabolic pathways are similar in all organisms. Amoebas, horses, algae all use cellular respiration and have the same organelles and enzymes to facilitate these reactions. Fourth, within eukaryotes, many metabolic pathways occur in specific organelles, such as the mitochondria. And fifth, each pathway is controlled by enzymes that can be inhibited or activated. Within cells, energy transforming reactions are often coupled, meaning that energy that is released in an exergonic reaction is coupled to an energy requiring endergonic reaction. Energy released by one reaction will be fuel for another reaction. 2 coupling molecules that we will learn about in cellular respiration are ATP and NADH. Adenosine triphosphate, or ATP for short, is often called the energy currency in cells. Energy that is released by an exergonic reaction is stored in the bonds of ATP. When ATP is spent and energy is released, ATP becomes ADP, adenosine diphosphate. Free energy is released from ATP to drive endergonic reactions. Figure 6.1 illustrates a transfer of energy from an exergonic reaction to an endergonic reaction. Energy that comes from an exergonic reaction combines with ADP to make ATP. Energy that is released from ATP to make ADP will go into an endergonic reaction. This is illustrating that the 2 reactions are coupled. You cannot have one without the other. Figure 6.2 illustrates a picture of ATP. ATP is composed of adenine and 3 phosphate groups. When ATP is hydrolyzed and loses energy, it will lose a phosphate group to become ADP. As ADP, it has no energy, but once it regains a phosphate group, it becomes ATP, it will have energy again. Hydrolysis of ATP is exergonic, meaning that energy is released. As ATP becomes ADP, free energy is released to contribute to another reaction. In ATP, the free energy in the bonds between phosphate groups is higher than it is in the bonds of ADP. Energy can also be transferred in the movement of electrons in reduction-oxidation reactions, or commonly called redox reactions. A reduction is the gain of one or more electrons, while an oxidation is the loss of one or more electrons. Students oftentimes get the two confused, as a reduction is actually a gain of electrons, not the number of electrons being reduced. Reduction refers to the reduction of an atom's positive charge. The acronym OIL-RIG sometimes helps. It stands for oxidation, it loses; reduction, it gains. So remember, reduction is the gain of electrons, while oxidation is the loss of electrons. Oxidation and reduction always occur together, as we will see later with cellular respiration. You can see that compound B is oxidized at first, meaning it has no electrons, but gains electrons from compound A. When compound B gains those electrons, it now is reduced, while compound A is oxidized since it has no more electrons. Also, when we talk about redox reactions, it's useful to think about it as the gain or loss of hydrogen atoms. This will become important later on in the lecture. The transfer of a hydrogen atom involves the transfer of electrons with it. As you may recall, a hydrogen atom contains one proton and one electron. So when a molecule loses a hydrogen atom, it becomes oxidized. When a molecule gains a hydrogen atom, it becomes reduced. The more reduced a molecule becomes, the more electrons it gains. This means that there is more energy stored in the reduced molecule's bonds. Redox reactions involve the transfer of energy in the form of electrons. Energy from reducing agents become oxidized, thus losing an electron, as it is transferred to a reduced product, which now has more energy at the end of the reaction. Figure 6.3 is a diagram of methane on the left, with carbon dioxide on the right. As methane is oxidized, it will lose electrons and become carbon dioxide. Methane has more energy than carbon dioxide because methane has more electrons in its bonds. Coenzyme NAD is a key electron carrier in redox reactions. It is important in transfer of electrons in cellular respiration. NAD+ is oxidized, meaning that it has less electrons, less energy. NADH is reduced and has more electrons, more energy. Remember we mentioned how hydrogen atoms can indicate if a molecule is oxidized or reduced? If it gains a hydrogen atom, as in the case of NADH, it becomes reduced. The reduction of NAD+ to NADH is endergonic, meaning that the molecule gains energy. The oxidation of NADH to NAD+ is exergonic, meaning the molecule loses energy. Figure 6.4 illustrates the oxidation reduction of NAD+ and NADH. Using the example on the slide, as NAD+ is reduced, it gains a hydrogen atom from the molecule A, thus oxidizing molecule A and becoming NADH. As NADH is oxidized, gives its hydrogen atom to molecule B, becoming NAD+, thus reducing that molecule B. Energy is released in catabolism by oxidation and trapped by reduction coenzymes such as NADH. Energy for anabolic processes where molecules are being made is supplied by ATP. Most energy releasing reactions produce NADH. Most energy consuming reactions require ATP. Oxidative phosphorylation transfers energy from NADH to ATP, which is the final step in cellular respiration. As we will learn, the ultimate role of NADH is to transfer energy to produce ATP. Okay, we went through a lot in the last 20 slides. Maybe you need to take a break to let it all sink in. Take your best shot at these progress check questions and continue the lecture when you are good and ready. Alright, with the background on redox reactions and NADH out of the way, it's finally time to dive deeper into cellular respiration. In a nutshell, cellular respiration is a set of metabolic reactions used by cells to harvest energy from food. A lot of energy is released when reduced molecules become fully oxidized, producing carbon dioxide in the process. Oxidation occurs in a series of steps, allowing the cell to harvest about 34% of the energy that is released. In living organisms, the energy released from glucose is released in a series of small steps, as you can see in figure 6.5. The same energy can be released by using heat, but the activation energy is higher, as you can see on the right. The energy being harvested wouldn't be as controlled, and plus you run the risk of catching on fire. Catabolism of glucose under aerobic conditions occur in three linked pathways: glycolysis, pyruvate oxidation, and the citric acid cycle. Glycolysis is where glucose is converted into pyruvate. Pyruvate oxidation is where pyruvate is oxidized to acetyl coenzyme A. And then the citric acid cycle takes the acetyl coenzyme A, is oxidized to form carbon dioxide. We will learn more about these steps in detail coming up. Figure 6.6 shows the step of cellular respiration in order. As I mentioned before, we will go through these. This is a good roadmap of which step comes where. As you will see in these reactions that we will go over, many of them have many steps. Main ideas I want you to take away for each of these steps is, what is the main product going into the reaction, and what are the end products of these reactions? Are they waste, such as carbon dioxide, or products that go into the next reaction, such as NADH? As we go through these reactions, we are looking at glucose and how it is broken down into products that are reduced and oxidized. The main goal is to take glucose, the energy in the bonds of glucose, to produce ATP. The first step in the breakdown of glucose is glycolysis. It occurs in a series of 10 reactions, and occurs within the cytosol of the cell. The final product of glycolysis are 2 molecules of pyruvate, which will go into the next reaction, 2 molecules of ATP, and 2 molecules of NADH. Figure 6.7 shows glucose at the start of the reaction. ATP is needed to get it started. As you can see on this slide, glucose is broken down further within those 10 steps of glycolysis. As glucose is oxidized, energy from its bonds produce ATP and NADH. But the important molecule at the end of glycolysis is pyruvate, because this will be a key molecule in the next reaction. As mentioned earlier, you do not need to know all the steps of glycolysis, but know what goes in and what comes out at the end of these reactions. Pyruvate oxidation occurs in the mitochondria within eukaryotes. It produces carbon dioxide and acetate, which is bound to form acetyl coenzyme A. Also, NAD is reduced to NADH because of pyruvate being oxidized. You can see here that pyruvate is oxidized by NAD+, becoming NADH, producing carbon dioxide and acetyl coenzyme A. Acetyl coenzyme A will be important in the next reaction. The next step is the citric acid cycle. It is a series of 8 reactions and occurs within the mitochondria. It operates twice for every glucose molecule that enters glycolysis, since glycolysis produces 2 molecules of pyruvate. Acetyl coenzyme A donates an acetyl group to the molecule called oxaloacetate. The oxaloacetate is regenerated in the last step of the cycle so that it may start again. Main products of the citric acid cycle are carbon dioxide, NADH, and FADH2. FADH2 is a similar electron-carrying molecule to NADH. Figure 6.8 shows a diagram of citric acid cycle. The diagram simplifies the citric acid cycle for us. You can see that NADH, FADH2, and CO2 are produced. The NADH and FADH2 will be important for the next step since they carry energy. Also, it is important that oxaloacetate is regenerated in the last step so that the cycle may continue. This slide shows us how oxaloacetate is regenerated. You don't have to know how it is regenerated. Just know that it is, because the oxaloacetate binds with the acetyl group from the acetyl coenzyme A, produced from the oxidation of pyruvate. Alright, we're getting to the end, I promise. Next step is called the transport chain. The NADH and FADH2 produced from the previous reactions contain energy. That energy from those molecules will produce ATP by oxidative phosphorylation. NADH will donate its electrons in an oxidation reaction, which is used to actively transport protons of hydrogen across the inner mitochondrial membrane, resulting in a proton gradient. The diffusion of the protons across the membrane then drives the synthesis of ATP. When NADH is oxidized to NAD+, oxygen is reduced to water. This occurs because the electron is donated from NADH to create the hydrogen proton gradient. The electron is transported through a series of proton carriers in what is called the respiratory chain. The primary role of oxygen in cells, and the main reason why we breathe oxygen, is because oxygen is the final electron acceptor and becomes reduced. And when it becomes reduced, it forms water. Without oxygen, electrons would build up in our cells, causing problems. This is a diagram of the inner membrane of mitochondria, where the transport chain is located. NADH and FADH2 produced from the citric acid cycle will donate their electrons to this chain. As this happens, hydrogen ions are pumped to one side of the membrane, producing a gradient. As hydrogen ions come back across the membrane, ATP is produced. There is a video posted on Canvas that illustrates this, showing the hydrogen protons being moved from one side of the membrane to the other, with the electron being passed from carrier protein to carrier protein. I highly recommend that you view that video. In electron transport, the electrons from the oxidation of NADH and FADH2 pass from one carrier protein to another in the chain. Energy is released from the NADH and FADH2 as they are reduced. This released energy from the electrons actively transport hydrogen ions across the membrane, making a concentration gradient. The hydrogen ions enter back through the cell membrane through the enzyme ATP synthase. As the hydrogen ions cross through the membrane, ATP synthase will turn ADP into ATP. So this is the reason why we have a hydrogen proton gradient. Without that hydrogen proton gradient created by NADH and FADH2, we would not get the formation of ATP. Figure 6.10 shows carrier proteins moving hydrogen ions to one side of the membrane, making a concentration gradient. Those hydrogen ions move through ATP synthase. ADP is combined with the phosphate to produce ATP by chemiosmosis. Chemiosmosis is just the movement of ions across the membrane from high to low concentration, which is occurring within the mitochondria. The ATP synthase is converting the potential energy of the proton gradient to produce chemical energy in ATP. ATP synthase structure is similar in all organisms. Prokaryotes, since they do not have mitochondria, use their cell membrane for the proton gradient. In eukaryotes, ATP is synthesized in the mitochondria using chemiosmosis. Alright, that was pretty self-involved. We went through a lot of material in the first half of this chapter. Please take your time to read through the book. Also watch the videos that I've posted on Canvas. It is important to understand this, as the next lecture will build upon what we have learned here. I know we went through a lot, and cellular respiration can be a little overwhelming, so I recommend just break it down step by step and learn what materials go into each reaction and what materials come out, and ultimately, hopefully, that will help you understand what is going on. And please, if you have any questions about any step of cellular respiration, please do not hesitate to email me or contact me. Thank you for listening to this lecture, and have a good rest of your day.